Yes, I'm speaking. You can hear me? Thank you very much, Prof. Ralph, for the kind introduction. I would like to start by wishing you all a good day first, of course, and thanking the African Society of Human Genetics and congratulating them on a successful event and for having me presenting their work of the Genodermatosis team in the National Research Center here. If you would allow me a minute first to tribute my late professor, Tanzania, who came back to Egypt at the highest time of brain drain from Hopkins to establish genetics in Egypt, and I might say in Africa and the Arab world. There are no words who can express the loss nor the gain of having her or my mentor, my teacher, and may Allah beautiful soul in eternal peace. Back to the Genodermatosis. I would start by speaking of generally what endogamy is the in marriage or the in clan or in cultural group marriage. However, in clinical genetics, it is the union between two individuals who are second cousins or closest, or who are having a coefficient of inbreeding that is equal to or higher than 0.0156, where this coefficient of inbreeding measures the proportion of loci the offspring is expected to have or to inherit as identical gene copies from both his. Why is consanguineous marriage favored? It's all mainly social because they think consanguineous marriage could enforce stability. There is higher compatibility between the husband and wife if they're belonging to the same family, or because the couple and the family members would feel more compatible, or because marrying within the family would reduce the possibilities of uncertainty, both on health and wealth matters, or to transmit the cultural values or maintain the cultural continuity. It's cheaper to marry within the family, and moreover, wealthy landlords may prefer to keep their properties within the family. So it's either health, culture, ethnicity. However, it started by the church, by royalty. The church, the Catholic Church, prohibited the royal to mix with the commoners, and they have to marry maximum up to the fourth degree relatives, as it was extended to the seventh, and then it either you defy the church to marry commoners or change these rules. Speaking of religion, something that the Catholic Church inhibits still the marriage of consanguineous marriage, I mean, of second cousins marriage. But this nice slide I found from a friend, it's only prevented by Indo-European and Sikh. Otherwise, there is no religion that prohibits consanguineous marriage. So, speaking of consanguinity from the beneficial point of view, they think that segregation helps groups or communities resist integrating or merging with other groups or cultures who do not share the same beliefs or resources, and help minority cultures to survive over a long period of time, encourages group unity and control over resources needed in order for the group or clan. However, this is speaking from the biological point of view, there might be a genetic disease that would wipe the whole of this clan, the whole population. So we have to weigh the biological disadvantages of consanguinity versus what they think is the cultural. It's very difficult to estimate how consanguineous we are, because at the very early years of human existence, nobody knows what was the state of inbreeding, and thinking of how was small the group of the size of the persons who had to make definitely intrafamilial reproduction occurred, with resultant very high levels of consanguinity. Still, in the recent times, consanguinity is thought to be very high in Africa. However, there are no certain numbers speaking of the Central or Southern Africa up to the reviews of the literature I've been searching. However, consanguinity is thought to be very high in Northern Africa, the Middle East, and Eastern and Southern Asia. And no matter the who think they have lots of homozygous, they share lots of homozygous loci because they have a long-standing history of consanguinity among them. And WHO report about the Eastern Mediterranean region, they reported a very high incidence of genetic disorders that are related to consanguinity, as is indicated in this green area, among, of course, other causes of consanguinity within the Eastern Mediterranean region. And these kind of pedigrees are so in our clinic, either with so many disorders and very high consanguineous and interrelated pedigrees like this one, even if it doesn't have any genetic disorders, it's very challenging to the public health providers because, as I mentioned in the previous map, nobody knows how consanguineous we are. And when they come to the public health provider or to the health provider services for premarital or preconceptional counseling or to the genetics clinic, they're asking, they wanted to have a healthy offspring. And it's very difficult to warranty such a counseling in our background. However, the sure thing is mutations that are not lethal, a reservoir for genetic diseases. And from here, we go to Genodermatosis clinic. I presented almost the same presentation, or I was asked to present the same presentation this year. Last year, when I spoke about Genodermatosis in the same context, I went through hundreds of cases who have been frequenting our clinic. But this year, I thought I might concentrate or focus on genodermatosis that are having high incidence of autosomal recessive inheritance, in spite of maybe some of them are not actually autosomal recessive. The genodermatosis cases frequenting our clinic have a high incidence of consanguinity reaching up to almost 80%, and a percentage of similarly affected SIBs within the families frequenting the clinic reaches up to 50% who are having either similarly affected or having another genetic disorder who inherited either within the SIBship or the same SIB even, or within other family members. The first disorder I will discuss was xeroderma It's an example of autosomal. We have lots of cases frequenting this clinic. Types, and we discovered variable genotypes too. We started by studying them through sister chromatid exchange in trial to correlate it to phenotype successfully in most of the cases. And then we started working on introduction of targeted sequencing, Sanger sequencing, where we detected several mutations. And these mutations, while reporting them, we by Sonia Abdelhaq. We attributed this to our common ancestors, particularly during the Fatimide period. And our student Iman Dr. Iman during her master's degree, she continued the work through studying 40 other xeroderma pigmentosum patients through sequencing XPA and XPC genes directed or based on guided by clinical findings. She could characterize all the mutations. And while doing this, she found that among our 13 pathogenic XP detected mutations, some of them were confined to Egyptians and were thought to be founder mutations, and others, when we trace them, they might indicate kind of migratory flows, such as the C1643, 1644 deletion, which is very common. It was reported in 74% of Maghreb populations, as they thought to be arising from Morocco and Tunisia, and then migrated through 20 to 50, 1250 to 1500 years ago. That's to say, during the Saracens or the age of the Islamic invasion of Northern Europe to Spain and Italy and Egypt and Morocco, and Turkey. Also, they were found in Libya and in Algeria, and that's why they were thought to be of Mediterranean. They're named now as a Mediterranean mutation. The second mutation was the XPC. It's called XPC. Oh, sorry, I cannot see it. XPC 2251. This mutation arises in the Eastern Africa, in the Comoro Island, and in Mozambique, and then from there it went to across the Red Sea to the Arabian Peninsula and to southeast of Pakistan, and then another new record found it in Brazil, where a study found it in 47% of their XP patients, and the only explanation was that it had passed through the slave trade from Portugal to Brazil. A third mutation was the mutation that was found in Italy and in Turkey. And this mutation was thought to have been transmitted to Egypt during the Roman or Ottoman Empire and the invasion of Egypt. The last mutation is a very rare mutation that was reported only in one study in Palestine, and it was related to the Sanctus Cacchione, and was reported in Brazil once, and it was with milder clinical phenotype, and now found in Egypt. And again, we thought it started here and then migrated with the slave trade to Brazil. The co-evolution and study of migratory flows might explain the common ancestors or explain the diverse genetic mutations we found in this part of the world. The second is non-syndromic ichthyosis, and non-syndromic ichthyosis are a very heterogeneous group of disorders. They're very phenotypically mild, very heterogeneous, ranging from very cases to severely affected lethal types, and they were all 55 studied patients were descending from autosomal recessive consanguineous pedigrees, except for one who had a GJB4 mutation, that is an autosomal dominant erythrokeratoderma varialis, and this was the only autosomal dominant case inherited or present in our study. The rest of the mutations were found mainly in 76% in the TGM1 gene, which is the case in most of the autosomal recessive ichthyosis cases. Speaking of, putting them as autosomal recessive because they're mainly coming from autosomal recessive pedigrees. Sorry for an adhan. It will take a second. Sorry again for the interruption. The stars here marks the mutations because the novel mutation that were not added here because she did not report them yet. This was done by Nesma during her postdoc study. And the third group of patients are the epidermolysis bullosa patients. Epidermolysis bullosa are quite heterogeneous. They represent about more than 30 or present more than 30. However, they are mainly grouped into four major groups: EB simplex, junctional, dystrophic, and the very rare Kindler syndrome. And this grouping depends on or is based upon the plane of cleavage within the skin that reflects underlying molecular pathology. We started by targeted sequencing, trying to target the most commonly reported mutations to be met by failure and failure and failure. When we started whole exome sequencing, we discovered why we failed, because among the 22 patients, we found 20 pathogenic variants. Not a single patient shared the same mutation like the second one. If the only 3 patients who shared the same mutation were the Kindler, who is caused by one The first group was the epidermolysis bullosa simplex patients. We had 3 patients, and maybe there's supposed to be much more than that. Maybe because in the low-income settings, people do not seek medical advice unless they have a devastating disorder. However, those 3 were not phenotypically mild, as should be the simplex cases. They were caused by 3 different mutations in 3 different genes. The 3 of them were novel mutations. And none of them was in the keratin 14 gene, which is very famous to cause epidermolysis bullosa simplex. One of the cases, the one case that was harboring the keratin 5 was the mildest of them all, except although the keratin 5 is reported to be lethal early in life. And we have one who has a plectin gene mutation, and she presented to the clinic actually with muscle dystrophy, and on examination we find that she's having a bullous lesion, and she was found to have epidermolysis bullosa. The second group is the junctional epidermolysis bullosa, and this is sorry, I forgot to add an important part. All the 3 simplex cases were autosomal recessive, although it's very rare to find autosomal recessive cases among epidermolysis bullosa simplex. They're usually inherited in an autosomal dominant matter. The second group of epidermolysis bullosa, or the junctional group, it's usual for the junctional to be inherited as autosomal recessive, and of course all our cases were presented through autosomal recessive pedigrees. 4 of mutations were novel and the rest were previously reported, and mutations were detected in the COL17A and LAMA3 and plectin gene again. However, none of the mutations were present in the LAB-B3, which is known to cause about 50% of the junctional epidermolysis bullosa cases in the literature. The only genopheno findings that we could find in the epidermolysis bullosa is this animal pitting and hypoplasia that is reported with this mutation. And otherwise, there was no genotype-phenotype correlation whatsoever, and there was marked genotypic and phenotypic heterogeneity among the cases. The interesting finding is the presence of mutations, which is not a common feature generally in epidermolysis bullosa. And it's either due to co-inheritance of 2 epidermolysis bullosa genes, and in this case it would act as maybe a phenotypic modifier, or 2 genes that are not actually related or associated with epidermolysis bullosa, but might be associated or affecting an EB protein. For example, somewhere in the locus control region or somewhere like that. However, among the 22 studied cases, we detected digenic inheritance in 4 patients, 3 belonging to the junctional epidermolysis bullosa, and one case belonging to the epidermolysis bullosa dystrophic group. An interesting finding too is that one of the parents was having mild manifestations of the disorder, although she was having one gene. She was having a heterozygous mutation, heterozygous mutation. And reviewing the literature, we thought it might be due to post-zygotic mosaicism for a disease-causing variant that might explain the milder phenotype in the parent or of this severe cases. All revertant mosaicism was improved mechanical instability and spontaneous healing. Of course, the majority are epidermolysis bullosa dystrophica, and where 10 of the 22 cases were belonging to this group. We reported here 7 novel mutations, and 8 of the 10 were descending from autosomal recessive, although the majority of epidermolysis bullosa dystrophica are inherited in a dominant matter. One of them was the case that is co-inheriting the plectin gene, and he was not presenting with any signs of muscle dystrophy. Again, the previous explanations might be applied to this absence of dystrophic manifestations, although the gene is known to cause epidermolysis bullosa with muscle dystrophy. The fourth type is the Kindler. 3 cases presented as Kindler. They were classic Kindler cases with acral affection, and only one of them have the tongue hemangioma, and they have previously reported mutation within the known gene FERMT1, and they all were, of course, autosomal recessive. This case of the Fraser syndrome case was referred to the clinic as suffering of biotin deficiency case, and when we couldn't find any mutation, we sent her to whole exome sequencing to detect a GRIP1 gene mutation, and she was found to be Fraser syndrome. Although clinically she doesn't have the triad of Fraser syndrome, still she's having hypogenitalism, which is one of the manifestations of Fraser syndrome. And she's having he was short and microcephalic and having this hairy mole on his back. That is, but if we're having a molecular diagnosis, it is the only 100% sure diagnosis. Sintani, in her study in 2012, concluded that in a highly consanguineous population, you would find high consanguinity rates even among autosomal dominant and X-linked disorders, sporadic chromosomal and others. And this is the case in the coming group of patients I'm presenting. I'm going to present the work of Nahla in her PhD thesis. She was working on neurofibromatosis, and this busy slide she always used because it concludes how confused she was when she started studying such a big gene consisting of 60 exons with lots of pseudogenes and overlapping transcripts, and she had only her Sanger sequencer to work with. She had, of course, to do RNA, cDNA to avoid the pseudogenes, and she confirmed her results by MLPA. We were faced with 45% consanguinity among this autosomal dominant studied pedigrees, and when we did some cytogenetic analysis for some of the cases because they had very unusual presentations, we detect chromosome 17 deletions in 27 cases of the NF1 of 40 cases. And these cases, when they did further analysis, they thought to be having some P53 anomalies, and they were thought to be prone to develop malignancy, and they are under close follow-up now. And this family, so co-inheritance, we don't know what that is. Is it co-inheritance of some progeroid manifestations, or is it some new manifestation of neurofibromatosis, because it is seen in the boys who are affected with neurofibromatosis and in their father who transmitted the disorder to them. These are the chromatograms, and these are the mutations that could be detected. The novel mutations are in the red arrows. After doing the RNA, cDNA, 4 of the patients Nahla studied, we could not detect any anomaly, any mutation, and we thought that we reached out actually for Necker in Paris because we're in a collaboration, sort of collaborative relationship with them. We're trying to do a genome sequencing or some kind of long read sequencing, because neurofibromatosis is known for the in-depth up to maybe 3,000 sometimes base pairs intronic mutations to try and find what the cause is. The interesting thing is that, although of course intrafamilial heterogeneity is something normal among autosomal dominantly inherited disorders, we introduced laryngoscopic services to our genodermatosis patients, and during the study of the PhD of Dr. Muhammad here, he found this pedigree. The mother is having mild manifestations who increased dramatically on pregnancy, and the first daughter has a little bit more manifestations because she started manifested early in childhood. while the third one is having here occipital plexiform neuro. And more importantly, she's having a laryngeal plexiform neurofibroma since birth. We sent her for surgical removal. She has unilateral vocal fold paralysis, and she's being followed up. Now, family studied by Muhammad. Again, the mother started a progressive course, which increased with pregnancy. The first daughter manifested very early since birth, and the third daughter was having a serious serious pulsatile pharyngeal wall mass, which could be an internal carotid, either aberrant internal carotid artery or carotid aneurysm, and in both cases this is life-threatening, and she had to go she was referred for vascular surgical intervention. And she was also having a very, very rare finding, which is a fibroma at the tongue, or called second tongue, at the floor of the mouth. So it seems that we would recommend the introduction examination in genodermatosis clinics. The fifth group, ectodermal dysplasia, is an example of X-linked inheritance. We studied 45 families suffering ectodermal dysplasia. 28 of them were X-linked as by clinical examination, and dental, of course, the mothers having some manifestations, and the rest were not molecularly analyzed autosomal recessive parts. The 28 families were descending from 20 unrelated pedigrees. 45% were consanguineous, and 10 of the 28 patients, which is very good finding, were harboring the same mutation. So we thought finally we can find a hotspot to target in the coming counseling sessions. However, we were faced with intrafamilial phenotypic variability, which is a great challenge, of course, in providing genetic counseling to patients. These are examples of the families ectodermal dysplasia patients. And these are, of course, the photos of the patients. All of them are sharing the sparse hair, the prominent lips because of the absence of the teeth, and of course the dental anomalies. And here you would see the arrows are pointing to heterozygous parents who are having no manifestations except for these missing teeth who were found on dental examination. Among the 13 found mutations, 6 were previously reported and 7 were novel mutations. And intrafamilial heterogeneity, as I mentioned, was a challenge in ectodermal dysplasia. For example, in this family, this daughter, the younger one, was having dental affection more than the older one, and she was also having nail affection while the older one didn't. And their mother was having some manifestations of ectodermal, mild manifestations of the disease. Although, of course, she's a heterozygous carrier. These are examples of the consanguineous families of ectodermal dysplasia patients who is an X-linked, these families are all harboring X-linked mutations. Again, this might be a proof of Prof. Timtami's study of the high instance of disorders, of different disorders, not only autosomal recessive, in societies with high consanguineous background. Another service with our patients at the Genodermatosis clinic is the psychological assessment and quality of life assessment. It's very important, of course, to help those patients because most of them are having severe psychological disturbances because of the visible nature of their disorders. Heba, in her PhD, is responsible for evaluating them from different concepts like their school performance, physical affection, psychological affection, among others. And we used to have a perfect psychiatrist, but she was taken from us by the private sector. Of course, it pays much better, but at least she still is in Egypt and we can ask for her help when needed. So Genodermatosis is very challenging. In spite of having the luxury of having now a grant that facilitated or helped us to help all those families by supplying, providing, I mean, next generation sequencing, still some of them we cannot help, like the neurofibromatosis cases, or like the, I forgot to mention, one of the ichthyosis, autosomal recessive ichthyosis, has a heterozygous we cannot find an explanation to, because we don't know. Deep intronic mutations were not previously described in ichthyosis, in the autosomal recessive ichthyosis, and this gene is reported to be of autosomal recessive nature inheritance. So lots of cases needs further explanation, and I would like to share with you these two cases, which I cannot. For years, we've been trying to find an explanation to them, while we could not. This pedigree is an ectodermal pedigree for this family. They presented with this daughter who has no hair, and she was having dystrophic nails and palmoplantar keratosis in a mild shape. And this is her sister. She's having dystrophic nails and she's having sparse hair. And this is the father having the full-blown picture like his first child, and the mother is completely normal, and the father is having marked palmoplantar keratosis and nail dystrophy, and yes, he was also having dental affection. And this is the father's sister having the full-blown picture of the disease again. And this is another son having severe palmoplantar keratosis and hair, and he was normal. And this is another son, he was having hearing affection with some dryness in the skin and contracture, but he has normal hair. Is it X-linked EDA, or is it an autosomal recessive EDAR? Coming to the pedigree. Okay, I'm finishing. So we cannot tell. The second case is a case who presented with fracture at an age of 1.5 years for both her arms. We diagnosed her Werner syndrome, but actually she didn't fit. On cytogenetics examination, we found lots of tetraploidy, and on fish she had P53 affection, and these are how thin her bones are. The nails introduced surgically for the recurrent fracture are almost as thin as the bones, and we could not reach any diagnosis for her too. In conclusion, consanguinity poses an important health burden, and still lots of people, particularly in low and middle income countries, think of it as a religious or important social habit. However, this favor intrafamiliar or population-based studies using pedigrees of consanguineous analysis to estimate the coefficient of inbreeding and to look for runs of homozygosity for gene mapping studies to be considered and associated an estimation of associated risks. Premarital genetic counseling has to be very important, and couples has to understand the importance of avoiding consanguinity to stop genetic disorders, and genome editing technologies is now hopefully being transferred to help affected families. I would thank you very much